All right, so now we left off with turning on the gene, getting to the point where we've made messenger RNA through transcription. Where we are now is we are talking about the processing of the messenger RNA before it is able to leave the nucleus. This does not happen in prokaryotes. They don't have these extra segments. They don't get processed. They don't get rearranged. They don't get spliced together. Whatever it is transcribed is exactly what gets translated in bacteria and prokaryotes. So, first off, caps and tails are added to the ends of the messenger RNA. So, if we're looking at this here, the cap is on the 5' end, and it's a modified guanine nucleotide. What does this look like? Guanine with 3 phosphates, that's GTP. That's the friendly cousin of ATP. And again, so we put a cap on the end. And then, as we are here, it's not, again, we don't just read this AUG and then that's it. There is an extra untranslated region. In addition to putting a cap on the end, we put a tail on the end. So 50 to 250 extra adenines, nucleotides get added on here, this poly A tail. We throw a whole bunch of A's on the end and we have the actual coding segment, but before we do anything, we first cap it. Again, it helps it leave, it protects it from degradation, because if something's going to get damaged here, if you protect the ends of the messenger RNA, that reduces the possibility of actually messing up the code for the protein. These caps and tails are also thought to help in binding to the ribosomes once it actually does leave. But once we put the caps and tails on, we're then going to splice out the introns. So they have this spliceosome protein, and this is going to grab on. And again, part of this spliceosome does involve protein, but it also involves very specialized, again, RNA. And RNA comes from DNA. So even if the DNA in our code is not coding specifically for proteins, because we often think of genes as the genes for proteins, we have all of this excess, you know, what's thought of as junk DNA. That junk DNA might be making some of these other forms of RNA, which add to the whole process in eukaryotes of making things a little bit more complicated and being able to not have quite as many genes, but turn those genes into ultimately different transcripts to make different types of proteins. So introns and exons, people can get these mixed up. Exons, exit, and express. So we are snipping out the introns and we are putting together the exons. So again, we cap it and then we cut out the introns and we end up with a transcript. Okay, so with a cap and the messenger RNA. So the average length of a transcription unit on the DNA is about 8,000 nucleotides long, which makes the average messenger RNA, once you take out introns, of 1,200 nucleotides. So that's the average size, and then those 1,200, make an average size protein of about 400 amino acids. So we have our messenger RNA. It leaves the nucleus, it finds a ribosome, and it is translated. So again, messenger RNA does not just make one protein and then it's done. A lot of ribosomes are hooking on to that messenger RNA. And as it feeds through 5 towards 3, we are adding amino acids with transfer RNA, reading the triplet codons, and we continue to build these growing polypeptide chains. And then there's these chaperone proteins which begin the folding. And they do fold because certain amino acids are attracted to other amino acids. Again, if you change the sequence of which amino acids, you're going to change how it folds up because this location might be attracted to that location and cause it to fold. So these chaperone proteins help in the initial folding of the polypeptide chain. And then there are another type of thing here called a chaperonin. So we can have this polypeptide that's folded up, but then it actually kind of goes into this cylinder, and this chaperonin ultimately makes it the proper shape, the correctly folded version of the protein, which starts making it functional. So again, different proteins have different shapes, and that's what makes them so unique and so versatile. And that's how we can get hormones and receptors and enzymes and muscle filaments and you know, cytoskeleton proteins and motor proteins and pumps and channels and antibodies and different types of connective tissue fibers. We can have all of these tremendously different amounts of proteins because you can assemble the DNA in different orders and then get different sequences of amino acids, and those different sequences can fold differently and you have different shapes. So, the next question is, humans only have twice as many genes as a fruit fly, but we're a bit more complicated than a fruit fly. But we only have twice as many genes. How in the world does that happen? If we have 20 to 25,000 genes that code for proteins, how in the world do we end up with so many different kinds of proteins? And this is a great little description here. When we talk about our proteome, we talk about our genome as the sequence of genes that we have, but the proteome is all of the different proteins that we have. And we have a transcriptome as well. So we can start with 20 to 25,000 genes. You can have 100,000 different transcripts, so these can be turned into different messenger RNA sequences. Those different messenger RNA sequences can then also be turned into finally different proteins, millions of different types of proteins. One way that we can get one gene into different transcripts is by changing the order that you put the exons together. So you have these spliceosomes, which again come from some of the other DNA, which is not actually, it's still making it's still making RNA, but it's maybe not making messenger RNA. The different sequences of the DNA part may not be genes, what we think of as messenger RNA, but they can be these things, and these can cause different sequences of exons to be connected. So when we're looking at um, this here, depending on how these are rearranged and reconnected, you can have one gene that makes one pre-messenger RNA, but you can reassemble that into different transcripts. You can have different, ultimately, recipes from the same initial recipe, depending on if you put this exon, then this exon, then this exon, or whether you go this exon and then this exon and then this exon. So again, one gene can make different transcripts. In addition, once we have that transcript coded into a protein or a polypeptide, we can fold it differently, but this would then be post-translational modifications. Translation is when we actually put the amino acids together. We can still modify it afterwards to go from 100,000 different transcripts coming towards the ribosome to a million different possible proteins, because we can fold them differently, we can attach different parts to them, Like this one, this glycoprotein got some sugars put onto it. And then even once it goes from here towards the um, Golgi apparatus, you can add so much and take away so much to the protein to change its function. So again, from the endoplasmic reticulum, this protein is put into a vesicle, and as it goes through the Golgi apparatus, we can do so many different things to it. We can turn this protein into a zymogen by attaching pieces which make it inactive until something else can remove that part and make it an actual functioning enzyme, for example. So again, we can get a tremendous amount of variation through the way that the RNA is processed, through the way the protein is altered post-translationally. But again, that's not the end of the story as far as genetics go. You can have different phenotypes, not just by modifying so many of those things, not just by turning things on differently, but we can also modify phenotypes by not turning things off. So once they're turned on, it's going to keep on, this messenger RNA is going to keep on making proteins, making proteins, making proteins, making proteins until you finally break down the messenger RNA. And then those proteins are going to exist until the proteins are actually broken down. So we continue to express that gene until we break down the gene. And that can happen through another form of RNA. So again, we have 25,000 genes, but we have so much DNA, extra DNA, which is more in charge of turning things on and turning things off. So we can take this other type of microRNA, chop that up by a different protein, and these little microRNA segments can ultimately bind onto messenger RNA, which would then interrupt the ability for the ribosome to read that messenger RNA. In addition, some microRNAs don't just block it, but they actually break down the messenger RNA, effectively turning off the gene. And then we're not going to turn that gene on again unless we go back to loosening up the DNA, and then by deacetylating it and then having the right transcription factors and the right activators in order to make that begin transcription. So we can turn it off through microRNA. In addition, the protein itself has to get broken down to turn off the genetic effects. If you leave it on for a longer period of time, you're going to have a different expression of that gene, which could ultimately affect the phenotype of an organism. So when we are ready to break down the protein and turn off its effects, the protein gets tagged for destruction by another protein. So these ubiquitins attach to the protein, which then tag it for the proteasome to break down that protein and chop it all up into peptides. And now these peptides can't do what the original protein was doing because they're no longer the same shape. So we've now turned everything off and stopped the expression. So we turn it on. We process the messenger RNA before it leaves. After it leaves, we make the protein. That protein is folded and can be modified and have different pieces turned onto it. So now we have fully expressed the gene, and we turn off expression by microRNA breaking down the messenger RNA or blocking the translation, or ultimately breaking down the protein. The last thing I want to just blow your mind with here is now let's think about mutations. Different things, and again, mutations happen more often randomly than through any types of mutagens or different things from the environment that can cause substitutions or additions or deletions, all of these things, and then those get changed and fixed. But you can have mutagens from the environment that can cause mutations, but what does that mutation actually do? Is the mutation a change in actually the genotype of a gene, or does that mutation affect the turning on or turning off of other genes? Holy cow, now we're getting a little bit crazy here because maybe the mutation isn't this, but the mutation's this, because this is a protein too. So if you mutate the gene that makes this protein, you're not going to break this down. If you mutate the gene that makes this protein, you're not going to break it down. If you mutate the gene that makes the microRNA, you're not turning it off. You're not breaking it down. You don't even have to ultimately change the original gene that we're talking about. We can change the turning off through mutation. We can change so many different things. Again, what is in charge of doing the splicing? Different types of RNA and different types of proteins. So again, if you mutate this DNA, you're going to change the expression of that transcription factor or that transcription. You're going to cause them, and then again, what is this? Protein. If you mutate the gene that's in charge of the chaperone or the chaperonins, you're going to change the way it's folded, which then changes the expression. And again, because proteins do so much, any point you change the proteins here, you're going to change the expression. You mutate this, you're going to change the expression. You mutate the activators, you're going to change the expression. You mutate any transcription factors, you're going to change the expression of this gene. So again, there's no end to how far you can go back. If you mutate the gene for acetylation, you're not going to unwind it and you can't express it. If you mutate the HDAC enzyme, because this is a protein too and this is a protein, then it's going to change what gets turned on and turned off. So when we start getting into mutations, it is incredibly complicated because you could mutate the histones. There's so many different things that you can mutate because proteins are not only are we making proteins, but proteins are running the making of other proteins. So any mutation on a DNA sequence is going to change this whole expression. And that's where we start getting, when we get into evolution and we start talking about so much of this other stuff or cancer, like it is an incredibly complicated process. So that's it. Eukaryotic gene expression, lots of different things going on, much more complicated than bacteria and prokaryotes, but it allows for so much extra variety.